Okay, I think we can start. I hope everybody hears me. Hello and welcome to the second workshop about BiofilmQ in this I2K. I'm Niklas, and I will guide you today through single-cell segmentation import into BiofilmQ. The current session, or in this session, also Eva is present, and Hannah. They both also developed BiofilmQ, or currently develop BiofilmQ, and are doing workshops in BiofilmQ. BiofilmQ, as a quick introduction, is a tool to analyze biofilms in a quantitative way, and it was developed by Raivo Hartmann, Hannah Jeckel, and Eric Yerli previously, and now Eva and me we help in maintaining it and giving workshops about it. Today's workshop will be about single segmentation with BiofilmQ. Here, let's first talk about this being a workshop. So this should be hands-on for you. So please just speak up when you have questions or if you want to say something, if something is unclear. And also you can do follow up on what we will do later with your own data if you have that. Otherwise, it should be hands-on also if you don't have your data yet. And the second important thing is this is with BiofilmQ, which I will come later to. BiofilmQ does not do the segmentation, it's for analyzing, but we will talk about this later. Yeah, before continuing to the single cell segmentation, let's quickly discuss what's going on tomorrow, because we have also a third BiofilmQ workshop on advanced topics, more advanced analysis and visualization. This will be tomorrow at the same time as now, and this workshop will be led by Hannah. So let's just get one step back and think about why do we need single cell segmentation in biofilms. Here, this is just one graph from our recent publication where we measured the division times of bacteria within biofilms, and for calculating these division times, we needed single cell segmentation to track single cells and see when they divide and thereby calculate these division times. And this comes with a challenge. This is single cell segmentation in three-dimensional dense biofilms. So we have very packed, dense biofilms, as you might also see here in this image. And we want to segment each single cell here. And now again, important, BiofilmQ imports single cell segmentations. So we will not talk about how BiofilmQ does the segmentation, but we will use other tools like StarDist, CellPose, or OmniPose that can do single cell segmentation in three-dimensional space, and that are optimized and better for biofilms. And then we will use these segmentations and import them into BiofilmQ, where we then can analyze the data. BiofilmQ then can analyze how long are the cells, how wide are they curved, what are their fluorescence properties, and what is the position within the biofilm of the single cell. So today we will have a small sample image, and we will use StarDist plus Napari and StarDist OPP, a tool that we developed for biofilm analysis or single cell segmentation. We will segment the sample image, then save these files and import them with BiofilmQ. And then in BiofilmQ, we will do a short analysis and talk about single cell properties that BiofilmQ can calculate now on this imported data. And then we will visualize a simple graph. Are there any questions so far? Otherwise, I will now start with the more hands-on part. For the hands-on part, we will, I hope people prepared themselves maybe. We have to install the Napari plugin for StarDist OPP, and we need to download a pre-trained model Can I get kind of a hands up who wants to really follow through the tutorial and who like prepared themselves? Or should I just go through what to do and no one will follow, but just watch what I'm doing? Is there anyone who really wants to follow while I'm doing the stuff? Okay, I don't see any hands up, so I'm just going to start. I will quickly, oh, there's someone in the chat. Okay. Most people just wrote, they just want to watch. Okay, so I have to stop for a second and then start screen sharing again. So let's start with Napari. I will start Napari in a console and Napari should quickly come up and you will see it. And furthermore, I will quickly go to this website. This is the website where we provide models or where we provide the StarDist OPP Napari plugin. And if you click on models, you see some zip files. These are pre-trained deep learning models that you can use to segment cells. I've downloaded this zip file already and extracted it also. So here is the zip file. You see my desktop, do you? Yes. Here's the zip file. I extracted it, and within the zip file, there's another folder named StarDist OPP7, and this here has configuration files and the information about the segmentation model, which we will use later. This is now Napari. I opened Napari already. And if you click on plugins, you can click on install, uninstall plugins. And in my case, in my case, I have already some plugins installed. And we are interested in this Napari merge StarDist masks plugin. So if you just type in Napari merge StarDist masks, you can find this plugin. And if it's not installed, this plugin would show up down here. And then you click here in blue on an install button. And once you click on this, the plugin will be in your Napari environment. I will close this again, and now we will start. We click on plugins and click on StarDist OPP. This is the plugin that we will be using. And this will take a little bit to open. And now this opens. Here on the right side, we see StarDist. This is the native StarDist plugin. And down here, we see our own StarDist OPP plugin, because we will go through using some StarDist functionality during this tutorial. Normally, you would just drag and drop now an image that you want to analyze into here. I will just open, if you just want to go along, you can click on Open Sample and click on StarDist OPP sample data. And then an image will open here. This is a biofilm, or a piece of a biofilm that we will analyze today. So we can scroll through here in the Z-axis, look at the data, and you see here the outline of cells. We can go to the Z. We can also look at this in 3 dimensions and see that this is a biofilm. What we already see here is the challenging part of segmenting biofilms is here in the bottom, the cells are brighter, and here in the top, the cells are less bright. Yes. But I will go back to this 2-dimensional view and now start with the segmentation. So first of all, we have to go through this part of the plugin. Here, we have to select the input image. There's currently only one input image open, so this is already pre-selected, and also the image axes are pre-selected. We have Z, Y, and X. The next thing that you have to do is click on Custom 2D 3D Model and choose the directory where the model files are saved. This is exactly the file, the folder that I showed previously, the extracted zip folder. So it's StarDist OPP7, StarDist OPP7 again, and then we click on Choose Folder. And then there is some more standard values. You don't need to change them. You can play around with them if you want to get a better segmentation, if your current segmentation doesn't fit you. But for now, we will just ignore these additional parameters and just click on Run. Now, Napari calculates, or StarDist calculates in the background some information, and this goes pretty rapidly. So now we have 3 new images that we can look at. I will not go through their what they mean. You should look up StarDist if you're interested in this, but yeah, we will not discuss this today. This is not a part of the talk today. So now we want to get the real segmentation. So the first thing is we have to copy this path above here and paste it into the first edit field down here. Next, we need to assign 2 other drop-down menus. You see that this drop-down menu is called dists for distances. So you have to use the following image, the StarDist distances image. It's available here in this drop-down menu. And similarly, here, this says props for probabilities, and you have to choose StarDist probability. Again, here we have a lot of additional parameters that are interesting, but for now we will not discuss them. And if you're interested in them, please let me know later and we can discuss a little bit about them if you have questions about them. Next, we click on Run. And now my computer will take, I don't know, 2 to 3 minutes to run and to create the segmentations, because yeah, this step just is a little bit more intensive computation. In the meanwhile, we will start BiofilmQ. And I will start BiofilmQ here as the exe file, so not within MATLAB, but I installed BiofilmQ as a separate software. Yes, BiofilmQ will now start. For those people that use BiofilmQ in this way, where they don't use it as MATLAB, but as a standalone software, you always have this field, this black console in the background, where you can also follow what BiofilmQ is currently doing. BiofilmQ takes some time currently to come up. Now it starts. So we now have to, or later on, have to import the files that we segmented into BiofilmQ. This means we have to import the raw image, the raw microscope image, and the segmentation mask. And normally, or sometimes you have different positions. So you have a great big experiment and you image one biofilm and another position of the biofilm and another position of the biofilm. And you do this also over time. And sometimes you also do this in different channels. So you have a green channel and a red channel. When we now later import data into BiofilmQ, BiofilmQ needs to know about, for each TIFF file that you want to import, which position does it belong to, which time point does it belong to. which information, which channel information does it belong to, and so on. So we will discuss this now. I have prepared the calculation that is done here in the background already, so we can continue on this. So BiofilmQ started, and we click on Browse. And now we go to the folder where we will later save our files. So this is the folder segmentation export. And within this folder, here you see this save folder in the Explorer. We have two files already. One file is named example, position 1, channel 1, time 1. And this means that this experiment is called example. This is a file that contains information about position 1, that contains information about channel 1 and about time point 1. And next to it is a file that is called example, position 1, channel 2, and time 1. So the same position, same time, but different channel informations. If we now go back, if we now go back to Napari is done already. The segmentation is done. We see the segmentation results overlaid over our original data. So this is our original data. We can scroll through again. And here now overlaid is the segmentation mask. If we click now on this segmentation mask, we can click here on the contour, and then we only see the outline of the segmentations. And when we scroll through, we now can better see that the segmentation is actually acceptable and good. So we see, for example, here's a cell, and we see that this is an outline, and it works to follow the single cells. We can also look at this in 3 dimensions, Napari, and see that there's many cells that were segmented. They have weird shapes, but overall they look nice, and we can continue to work with them. So now this is the segmentation mask, and BiofilmQ, when you want to import something into BiofilmQ by segmentation into BiofilmQ, you have to regard this segmentation mask as a separate channel, similar to having a fluorescence channel. So we will first save this file as channel 1, because this is our raw microscope image that we will also want to import into BiofilmQ. So we'll click on here, go on File, and click on Save Selected Layers. And now we will just overwrite these files that are previously prepared already. So remember, this is the raw microscope image. It's our example file. It's position 1, channel 1, and time 1. Channel 1, because we want to save the raw data as the microscope or the imaging channel 1. Click on this and save it, and yes, we will replace the file. Then we click on this layer in Napari, which is now our segmentation mask, and do the same. We click on File, Save Selected Layers, and now save it as channel 2. So it's the same position and time, and we regard it as a separate imaging channel, but we will come later to this. So we save it as channel 2 and click Yes. Now we can go back to BiofilmQ, and I will start again. So we have BiofilmQ here, and we click on Browse in order to open now this folder segmentation export, where we just saved our 2 files into. We click on Choose Folder, and now BiofilmQ analyzes this folder. And we get now this view where BiofilmQ tells us this is not a folder it can work on currently, because first we have to import some files. The BiofilmQ imports files based on certain patterns. So first we have a label, then we have a position denominator, then we have the channel denominator, and then we have a time point denominator. These can also be changed based on these regular expressions that you can find here. But if you don't care about this, it's better to just adapt your file names to have underscore position for the different positions of imaging or field of use on your imaging, underscore ch for the channel position, the channel number that you're using, and underscore time for the different time points. And if we click now on validate files, we see that BiofilmQ, based on this pattern of naming the files, sees two files. Example position 1, channel 1, time 1, the one that we saved, and the same file for channel 2. And we can scroll to the right to see what internal numbers they get assigned. So they are position 1, they are time point 1, and one of these files is for channel 1 and the other one is for channel 2. And with this validate files, it's good for you to just see which files would be imported based on these expressions that are given here. If you want to import two files, but only one of the files is listed here, then you know, okay, something is wrong and I have to check the names of these files and maybe adjust them. But we are good here, so we will now click on import custom TIFFs. This allows us now to import the files. There's no metadata currently associated to the files, so BiofilmQ pops up a warning about this. And for example, here, this is about time metadata, but this is not too much important for now. We'll just click one. And once the export is finished, BiofilmQ asks if it wants to switch to the different directory, because when importing data, BiofilmQ creates a separate folder where it copies the data, the imported images to, so that when BiofilmQ now works on these images, it will not destroy the original images, but will work on a separate folder. And it asks if we want to switch directly into this new folder. And we click yes. Now we are in this new folder and we see one file because we are currently selected. We're currently in fluorescence channel 1. We can click on show CStack. And again, similar to Napari, can scroll through our image. We can also click on auto view and open this view of our data. And we already readily see that there's something wrong or weird because this spacing here is too much. So our XY image is very small and our Z direction up here and up down here is pretty big. So we have to go back and change the scaling. It's currently set on 200 nanometers, but the data, when we acquired it, we use confocal images with 100 nanometers set spacing. So we will change this to 100. Click on update all images. And then we can have another look at the auto view. And now this is better and we can jump around and see how our image looks. This is good. So next, let's check fluorescence channel 2. Fluorescence channel 2 is now here misleading because it's not a fluorescence channel. It's what we exported as the segmentation map. So if we click again on auto view, we now see our segmentation where each cell is differently colored. Therefore, we see these colored cells. This is also good. Now we will go to this to these tabs here on the very right of BiofilmQ. We don't have to prepare our images because we're currently not analyzing single colony, or we don't have to align our image or align images over time. This would be done here. We are directly interested in segmenting these cells, but actually not in segmenting, but in importing a segmentation. We have to change back to fluorescence channel 1 first, because we are interested in segmenting fluorescence channel 1. And now we have this drop down here, this drop down menu here, saying thresholding and label image. And we click on label image. And then we have many possibilities similar to that what BiofilmQ segmentation with thresholding would also have. We have cropping, we have pre-processing, and we have denoising. Cropping and pre-processing are not so important. Denoising is sometimes important, but we will disable it now. So we will uncheck these two options because they will just take some time and we don't need them. This, if you had checked them, your image would be denoised, but this has no influence on the actual segmentation. It would just mean that you can remove maybe some background noise or get a denoised image for the processing, but normally you don't need this if you import segmentations. Then we click on label image, and here now we have to choose the channel that contains our original segmentation that was done previously. And we will click channel 2 in this drop-down menu because this is the channel where we saved our segmentation masks. Then there's also some post-processing and merge and transfer that we don't need in this case. And now we can click segment cells. And this is a rather fast task that's done already. So now BiofilmQ has imported the segmentation that we are interested in. If we click now on auto view, on overlay, to click on overlay, we see the imported segmentation. So now we see in grayscale the original raw image that came from the microscope in grayscale, and with red outlines we see the imported segmentation. So we can again go through here and look at the segmentation. But this should be now, or this is exactly the same segmentation that we've previously looked at in Apari. Maybe we can now look at something interesting again. We see down here the fluorescence intensity is somewhat high, it's pretty high, and up here the fluorescence intensity is low again. So we close this, we can actually leave it open, but we are now interested in quantifying this and making a graph out of this. We see this behavior that the fluorescence intensity is high below and lower in the higher set slices. So yeah, let's quantify this. For this, we go to parameter calculation. And here we now see all the parameters that can be calculated with BiofilmQ. I will focus on these parameters later again, but for now, let's just continue to make a nice plot as fast as possible. We are now interested in fluorescence properties. So we go here to fluorescence properties and check this box here. And then we have to go down here in the very bottom part of BiofilmQ and say which exact fluorescence property we want to calculate. We take from this drop-down menu here, that's named task, we take the mean intensity per object. So we are interested in the mean fluorescence intensity of an object. And we take channel 1 because this is our microscope image. And then we click add. And then we click calculate object parameters. And this is again rather fast. So we have calculated these object parameters already. And now we want to visualize them. Therefore, we go to the visualization tab here on the very left and have to click here on the very right, load data. So import now or load the data that has BiofilmQ has prepared for us. Anyway, we were interested in the intensity over the height of the biofilm. So we click on the intensity. Oh, sorry, no, we don't. Yeah, we can click here, but we want to have, so way to visualize this would be now a 2D plot where we have maybe the height on the Z axis, on the Y axis, so the Z height on the Y axis, and the intensity on the X axis. So we want to have a 2D scatter plot, so we click 2D XY scatter plot. And then we add the intensity, mean channel 1. This is the intensity of each object in channel 1 to the x-axis. And we add the distance from substrate to the y-axis. But this is now for each object that we segmented, we have mean intensity and its position or its distance from the substrate from the very bottom of this of the biofilm. And we will now plot each of them in a scatter plot by clicking here on plot, and then we get this graph where we say where we see exactly what we already saw by eye. This is at very high set positions. We have very low mean intensities, and if we now go down in the biofilm, we come to the lower parts of the biofilm, and the lower parts of the biofilm have high intensity. And leave this plot here. And some of you might not find this scatter plot very nice because it's a lot of scattering here. So maybe you want to make it nicer where you say, okay, you want to actually average these points, what's their standard deviation, average these points, what's the standard deviation, average these points, what's their standard deviation. And that's what we are now doing. So we will now plot on the z-axis, on the y-axis, the distance to surface, and then the x-axis, the x-axis, the distance to surface, and on the y-axis, the mean intensity. But in a slightly different plot, we use the 1.5D histogram. We take the distance from substrate on the x-axis and the intensity, the mean intensity on the y-axis. We take here a method called mean plus minus standard deviation, so calculates the mean of certain points, and we want to plot error bars, so set a checkbox here, and then we click plot. Now we see similarly nice plot. So for low distance to surface, we have high mean intensity, and when we go higher up in the set slice, we come to less mean intensity. I will now close these and move back from the visualization part, because we will now discuss some more properties that can be calculated with BiofilmQ, more single cell properties. Are there any? There's some message in the chat. I will quickly read this message. Hafiz, could you elaborate on your question a little bit?
Hi, yes.
Hi.
Because you showed at the beginning that you do the segmentation with NumPy and StarDist, because I do not have now currently with the PC. It was in somewhere else, but then it was really nice. But you showed that at the end you do somehow resegment. Did you resegment it again, or you just want to show that the BiofilmQ can do the similar segmentation?
No, it's not.
No.
BiofilmQ did not segment it. It was importing it. So is this your question? So we did not segment with BiofilmQ. We just imported the segmentation that was previously done in NumPy and StarDist.
But you do have a tool segmentation part of biofilm. You don't have, so you do
No.
need the Napari. Okay.
Exactly. Napari or whatever, like whichever tool you want to use that is very good for your data set, you can segment. And we,
Okay.
exactly.
All right. So. Okay. And a bit confused because you show also the red surrounding the cells. What are those? Are also from the Napari?
Yeah.
Okay.
So
all right.
Exactly. These are exactly the same surroundings that were done in Napari, but now imported into BiofilmQ.
Okay. Thank you.
Thank you. Any more questions for now? Okay. Then I will continue now with the single cell parameters that are available within BiofilmQ. And these start here with the, with the size, aspect ratio, and orientation by ellipsoidal fit. So for each, I will look at the overlay again. So we have here the overlay, and we have here each red outline circles one segmented cell. And we can now fit these cells by having an ellipsoidal fit and calculate then their aspect ratio. So for example, the cell, it will fit its length, calculate a proxy for its length, for its width, and also for its height or depth in a way, and then calculate the aspect ratios so that we can calculate how much longer is the cell than it is wide. So the aspect ratio. Then we can calculate the distance to nearest neighbor, which is kind of a measure for how dense is the biofilm in this area. And we also have the local density, which is also a measure for the density. Then there is intercell spacing, which is another measure for the density, but yeah, they are all calculated a little bit different. And it's again about how much, so it's different. It's about how much space is in between two cells. And furthermore, there's the alignments and nematic order parameter, which is calculating how much are two cells aligned to each other. So we have this cell, for example, here, and this cell here, and they both somewhat aligned into the same direction. But we have also sometimes cells where they are not so much aligned. For example, let's see, this cell here and this cell here, they are more kind of perpendicular to each other. And we sometimes are interested how much are cells aligned with one another or how much are they not aligned with one another. And this is something you can calculate here with the alignments and nematic order parameter. And that's the most interesting. Oh, we also have the convexity. I forgot that one. But with the convexity, it's just how in the context of fecal cholera, which is this biofilm, it's, for example, a measure of how curved is the shape, is the cell shape. Is it a rather straight cell or is it somewhat curved? In order to look at these parameters, we will now select some of them. The aspect ratio, for example, and the alignments and nematic order parameters. We will click them, check them in the boxes, and click again on calculate object parameters. This will now take a little bit longer. Have to wait until this is done. Okay. While we wait, are there any more questions? Because with this, we are actually through most of the part that we wanted to show. And we would be now open, or I would be open for any questions that you have or help. And as Eva and Hannah are here, we could also help you with any other questions about BiofilmQ, maybe not related directly to single cell segmentation, although single cell segmentation would be the topic of today's workshop. So we are open for any questions that you have. If there are no questions so far, I will just continue on visualizing some stuff. So we go back to visualization and click again load data to load the newly calculated parameters. And now we might be interested in, if we go back to the image processing, looking at the overlay, we might be interested in how much does this alignment with so we see some of these cells are aligned vertically, they point upwards. Can calculate now, is there some trend? Are the cells on the bottom more upwards aligned than the cells at the top here or not? So we go to visualization and we put the distance from substrate again on the y-axis, so it's up. We use a 2D XY scatter plot for now, and then we can take the alignment to the z-axis on the x-axis and plot this, and then we see that there's not a big difference. So this was a hypothesis that the data does not support. We can similarly also look at the aspect ratio length to width. Does the aspect ratio change? Are the cells at some position longer or shorter? Or do they have different aspect ratios? We change. Put this on the x-axis and look in the y-axis. And again, we don't see a clear dependence on the distance to surveys. But we can make one more sanity check, and this would be to look at a 1D histogram of the shape aspect ratio, just to make sure that we have somewhat working segmentation. And there we see that most of our cells have an aspect ratio of 2, which is pretty good because we know that Vibrio cholera, the bacteria that we imaged here, have an aspect ratio of 2. Okay, there's a question in the chat that I will read. How is the measurement of convexity affected by the segmentation approach? I am asking because StarDist assumes that objects to detect are convex. Therefore, I would expect the measurement to be biased. Have you compared the range of convexity estimated from the same sample but segmented with different methods? Okay. So, yeah, it's correct that StarDist assumes that the objects are star convex. However, here we are, so yeah, this is a kind of very big bound already on what objects can be detected, because they have to be in a way convex. However, we have used here StarDist OPP as an additional post-processing step that also allows to segment cells that are not convex, but somewhat curved. This is something we developed especially for Vibrio cholera biofilms, because with StarDist alone, we were not able to segment cells perfectly, but because the cells are curved, and with StarDist alone, this curvature could not be captured in the segmentation. Yeah, so the cells here could in principle be curved and have a weird or a different aspect ratio that is different from the convexity, from a perfectly convex aspect. Sorry, not aspect, from perfectly convex shape. Yeah. So, and yeah, we have not compared convexity estimates for different samples so far. I think or for different methods, segmentation methods. However, we have used these measurements here already to measure the convexity across species, and there it worked out. Okay. With showing you these additional measurements, I'm now at the end of what I planned for you, and I'm happy to take any more questions about anything regarding BiofilmQ or segmentation or StarDist or yes, so segmentations in biofilms. Hi, Michelle. Thank you for your question. I will read the question. I have a general question. Is it possible to evaluate co-localizations of two different channels, two different proteins in BiofilmQ? So this would be, yeah, a co-localization. I don't know if what I will show you now is really the perfect example for a co-localization measurement. But what you could do is having a segmentation of your cell and then go back and import two channels from a microscope and a third channel as your segmentation mask. So you would have 1, 2, and a third channel here. And then you would use, go to segmentation and go into channel 1, use the label image method, import the label for the channel 1, do the same for channel 2, and then you have two channels in BiofilmQ that are now that are segmented, and then you can go to merge and transfer the segmentation of channel 2 into channel 1, and then you have one channel with two separate segmentations, and then you can go into parameter calculation and click here on the fluorescence properties, and you can add the mean intensity per object for channel 1, and you can click on channel 2 and also click add. I cannot do this now, and then you will BiofilmQ will calculate for you for each object the intensity in first channel, for example in GFP channel, and the mean intensity in channel 2. And then if you plot them, you can see which objects have intensity in both of these channels high, or intensity where only GFP is present, or no mRuby, or both is present, or only mRuby is present, no GFP. Does this answer your question, or are you interested also in subcellular localization? I think you are not. Okay, any more questions? I will just sit here and wait for any more questions. Please just speak up if you want to, or write something into the chat. Hafiz had another question. Hafiz, do you want to directly ask the question via microphone? I think then it's easier to discuss.
Yes. Yeah, because in typical confocal microscopy, you do have like fluorescence. You do like say example of reporter genes, or you use certain kind of like fluorescence protein inside the cells. And we do also have a consideration of doing multi-species, like the question of co-localization. But one of the lights that we use in the detector is also the transmitted lights, because we can also look in the across in the Z stack of the images that we do also have the the transmitted light. It's not just specific on the fluorescence. Somehow, can we do extra or win any information from the transmitted light, or only BiofilmQ specifically collected the data from the fluorescence?
So is this, can you regard this as a separate is this channel? Is this also a three-dimensional information?
Yes, yes.
Then I would suggest just treating it as an additional fluorescence channel.
Okay.
I don't know what kind of the biological interpretation. I cannot tell you what it means, what the information is, but you can easily import it also as additional channel and then analyze it.
Yes.
Calculating all the fluorescence properties. Yes.
Okay, I will try that. Yeah, that's possible. Because BiofilmQ do have also microenvironments. It's not just the cells, it's also have a lot of metrics and so on. If you can see, like we did some treatments and so on, certain details, maybe I can't say, but we want to actually extract all the information that we can. So we do have a lot of transmitted lights, which is unused, but if you can somehow detect that in the depth or certain depth where the cells are maybe agglomerate or grow at certain height, it could be an important information for us. So so just treat it in extra channels and it should be okay. And the segmentation should be run with Napari and also StarDist like you suggested.
Yeah. So okay. So for the StarDist or the Napari plugin, what I would suggest is you use the plugin to play around.
Yes.
But once you I don't know how big your data set is, but if you have a big data set, I would maybe recommend automatizing your segmentation, but you can still use StarDist because it's in Python, it's easily written.
Yeah, we do have kind of big data set for pilot. We want to try for certain time points. Of course, we don't want to do for the whole time series will be big and will take long, a few hours. But if we can have like a pilot, a proof of concept would be nice.
Yeah. So the plugin is mainly mentioned as like just playing around with parameters.
Okay.
Trying out whatever works, and then once it works, you can run a big pipeline.
All right. Thank you.
Yeah. Thank you for your questions. Okay. Any more questions from someone? Hi. Hi, CY. This is a good question. Sometimes your image from above or like the Z direction is not directly. Sometimes it's inverse, that the Z direction, the offset direction. Bottom of the biofilm is the highest Z direction. In image preparation, there is in image preparation, there's the image alignment. Oh, sorry. Look. It's in segmentation pre-processing. You can say invert stack along C. This inverts the directionality of the Z-axis, and therefore you can then have the distance to surface or the surface of your biofilm at the top of the Z stack. Does this answer your question? Okay, so we are now kind of at the end of the workshop. I'm still here if people have questions. Happy to answer questions. Also Eva and Hannah, I think, are very happy to answer your questions. If you come up with questions further down the road, feel free to also write a message to us, for example, in the ImageSC forum, that most of you might know. So you can go to forum.imagesc and click on the community partners and click on BiofilmQ and just let us know about your problem. and we will be happy to answer your problem and help you with your problems. Or also, if you find something problematic, or like one of your workflows works but is tedious and not very smooth, we are also happy to work on BiofilmQ to make it more smooth for you. And yeah, with this workshop is ending. So if people don't have any more questions, I think you can leave. Otherwise, just stay and ask.